to present his slides.
I request all the participants to kindly mute yourself so that during the lecture of the resource person, he is not disturbed by the background noises. Please mute yourself, all the participants, students, please. Tulika, I think you may start now.
Okay, ma'am. Okay, ma'am. So, welcome everybody. And today is the second day of the 7-day workshop on recent trends in plant sciences. And yesterday we had a marvelous opening keynote lecture by Professor S.M. Paul Khurana. And today we have a great personality has graced this occasion, and I want to introduce him. And before that, I also want to welcome you all. And before the chief guest, I would like to say that our chief guest is Dr. Debashish Chakravarti. He's senior scientist in NBRI Lucknow and is heading the tissue culture department. Then I welcome our head of the department, Professor Pooja Singh ma'am. And she's really a person who takes things forward in a very humble way. So, ma'am, thank you for all the support in conducting this workshop. And then moving on, our colleagues have joined here, and also our former head of department, V.N. Pandey sir. I also welcome him. And then our colleagues, Dr. Deepa Shrivastava ma'am, Dr. Shweta Shekhar ma'am, Dr. Rakesh Pandey, Dr. Rajveer Singh. They all have joined us from their different labs. And then there are so many students of PhD and MSc. So welcome one and all. And now I will introduce our chief guest of this session. He is Dr. Debashish Chakravarti. Dr. Debashish has completed his PhD in Botany from the NBRI CSIR Lucknow, and he did his postdoctoral from the Chungbuk National University, South Korea, and CNRS Poitiers University, France. And now he's working as senior principal scientist in CSIR NBRI Lucknow, and he has guided till now 12 PhD students. He is the recipient of Gandhian Young Technology Innovation Award as a guide by the Vice President of India. He has published more than 130 papers in various reputed journals. He is editor of various journals, including the POLS1 and the Scientific Reports, which is of the Nature publishing company. He is working for a long time on the molecular arsenic metabolism in rice, identifying the genes and developing the desired gene, the transgenics. He is into editing genes, identifying the wild varieties in the rice germplasm. And it is a great area for this humankind because the public get affected by his research. Because we all know that we are rice-eating nation. We major parts eat rice, so it is a great. His research is a great thing on the society. In 2018, the Hindu paper, we all know, he quoted him as the top 20 scientists in India. Also, in vitro biology, he is one of the pioneer workers in the field of bioreactor technology. So we are very much happy that, sir, you have graced this occasion. And now, without losing any time, I want you to please unmute yourself and give your lecture. Thank you, sir.
Okay, thank you, and good afternoon to all of the faculty members as well as the students. And thank you, Dr. Tulika, for the nice words. So I just go directly to the presentation. Is it visible in PowerPoint?
Yes, sir. Yes, sir, it is visible.
And my voice is okay?
Yeah, yes.
Okay. So today what I will discuss, I will discuss the plant biotechnology and its application. I will start very rudimentary way that what is a biotechnology and other thing, and then we go for the application part and how it can be applied in our daily life. And also last of my presentation will be what NBRI is doing, because I think it is very important to know that what NBRI actually is doing, the other people, not me, other people are doing. And lastly, I will discuss some of my slides that what I am doing. So I think that in that way, I think you can enjoy, because there are a lot of students are there. So directly going into research will be not a good idea. So I request everybody to mute yourself so that no background noise will be available. Okay, so if we come into the first slide, you can find that there is a person who is a very famous, Steve Jobs, who said, I think the biggest innovation of the 21st century will be at the intersection of biology and technology, and a new era is beginning. And I'm sure with this last 1.5 years, we understand the importance of biology. We all are sitting at our home and waiting for vaccinate ourselves, and also waiting for when the institute will open for the students or ours also to start working. Not only that, there are a lot of populations are there who are not having their jobs. A lot of problems are there, and this is because of a small thing, COVID-19, and this is biology. So now the question is, what is biotechnology? Now the simplest definition of biotechnology is the applied biology. So in another way, you can say that the biotechnology is the use of biological process, organisms or systems to manufacture products intended to improve the quality of human life. This is very important. Those who are students, you should learn these things, that this is intended to improve the quality of human life. That means it is for the mankind. It's not like that. There are a lot of conspiracy theories coming right now that the coronavirus, is actually a product from the Wuhan lab, virological lab. So this is not a biotechnology. If it is true, I don't know it's true or not. There are some conspiracy theories coming out in the newspaper. So if it is true, then it is not a biotechnology, because it actually harmful for the human beings. So the products which is intended to improve the quality of human life, that is biotechnology. Yes, the Covishield is a biotechnology, product of a biotechnology, because it gives you the immunity. So in that way, you can define the definition of biotechnology. Now, you can just divide biotechnology in two areas. That is one ancient biotechnologies and another was in the modern biotechnology. Now, what is ancient biotechnology? Ancient biotechnologies are the biotechnologists who are the basically farmers and who are basically domesticated plants and animals using several techniques. And the modern biotechnology is nothing but a nowadays biotechnology that manipulates genetic information of organism, genetic engineering, like that. So now we come to the earliest example of biotechnology. Now, if you consider earliest example, then earliest example maybe you can find is 10,000 years ago, when our ancestors started keeping plants as a reliable source of food. Rice, barley, wheat, but they among the first domesticated plants. They used to domesticate them, those products. Also, they are domesticating sheep, goats, cows. These are the animals who are actually domesticated. Not only that, our ancestors also have the knowledge how to produce cheese, yogurt, dahi, bread, also make some alcoholic drinks. Even our lot of mythological books, for example, in Ramayana and Mahabharata, Somras is there. Somras is nothing but an alcoholic drink. So how you can make the alcohol if you don't have a knowledge of fermentation technology? So this type of technology already existed in the past. So the ancient biotechnology is the example of there. So now history of domestication started around 11,500 years ago in both areas. One is in the Fertile Crescent. Fertile Crescent is nothing but a delta, Nile Valley that is in Egypt, and Choga Golan that is in modern day Iran, where the wild barley, wheat, lentils are cultivated and domesticated forms of wheat appeared about 9,800 BC. So you can imagine how old people are they knew that how to domesticate plants and how to cultivate plants. And here in the left side, you can see a photograph from the pyramid.
पौधा के संरक्षण करेगा विश्वविद्यालय जितना मास्टर बने अब तनी कैमरा ऑन रखो जरा अंदर गलिया रहा है
All the participants kindly mute yourself because the resource person is getting disturbed. If you are not complying, all the participants, please mute yourself and please don't disturb the resource person, please. Yes, sir. Please continue, sir.
Yeah, thank you. So in my left hand side, you can see a photograph from a pyramid where you can find that they domesticated the animals and they use those animals for the cultivation purpose. So in in a pyramid in Egypt, 7,000, nearly 8,000 years ago, you can find that this kind of things are there. So they knew these things. And you will be very surprised to know that there are 7,000 years old jars of wine excavated in the Jargos mountain. Jargos mountains are basically in Iran, now on a display at the University of Pennsylvania. So you can understand that this type of fermentation technology was known by the people 1,000 years ago. So it's long before they knew. Fermentation technology in the biotechnology we used to read as a subject. But this subject is so old. That is very amazing. Now, the question is, why people began to settle down and become sedentary. Because in a civilization, for example, in case of Nile Delta civilization, or in the Iran, Chogagolan areas, the civilization, people used to settle down there. Now, why it is like that? So maybe in response to a population increase and increasing demand of food, so people cannot be moving from one place to another. If they become sedentary and they can restore the food, then they can have that food during the odd times, during the winter times or something like that. Shifts in climate, climatic shifts also another important thing which may be responsible for to settle down in a particular place. And domestication resulted in a food supply being greater in certain times of the year. That is also a very important aspect for the people who want to settle down. So what the people used to do is people used to collect the seeds of the wild plants for cultivations. For example, initial corn was very small and not juicy in nature. But these people, what the people used to do, they didn't know genetics. They didn't know anything about that. They used to cultivate those plants in a large area. And then they suddenly found that yes, among the population, there are some plants which is having a juicy kernel and a big kernel. So they choose those plants and then they cultivate those plants again. So for this type, you can see, I saw, this is the photograph that how corn is domesticated. This is the, initial one, and slowly it becomes juicy and long. So they didn't know genetics, they didn't know any other things, but with the basic idea, they cultivated and discovered the process. Now comes to the modern biotechnology. Now what is modern biotechnology? By the end of the 19th century, biotechnology was flourishing. Not only microorganisms being isolated and identified, but we know that Gregor Johann Mendel's work on genetics, especially the two theories, improves the breeding program, as well as the Pasteur, who discovered the pasteurization process, the microbial processes. That's also add on in the biotechnological process. 1943, the first direct evidence that DNA carries the genetic information appeared. In 1953, Watson and Crick produced their double helix model. And after that, there are several technology developed, like restriction endonuclease, like polymerase chain reaction machine, how to do it. So with this, all the new technologies is coming. The modern biotechnology began with their discovery. And also during the 60s, the tissue culture was flourishing. And from in India, Bhojwani, Dr. Bhojwani was a pioneer, you can say the father of plant tissue culture. And slowly, the tissue culture is one of the technique which has been used for the modern biotech programs. So with this way, the modern biotechnology was started. Now, the question is why we need a modern biotechnology? The thing is that after Green Revolution, we just use almost all the gene pools. I mean, it's very difficult nowadays that you can have the rice with improved nutrition, because there is no options left, because we used all the gene pools. Now gene pools are becoming very limited. So genetic variation, what we need is actually not there in the particular plants. But in another plants or in another things, it is there. So we wanted to just take from there and incorporate into our existing germplasm. So that's why nature doesn't contain all the genetic variation that human desires. Now, what is the present scenario? In the present scenario, the farmland is decreasing because of the urbanization, because of the soil erosion or soil conditions is getting worse. So farmland is decreasing. Population is increasing at a very alarming rate, and you know that. In India, it is right now 137 crore people are there. It's very difficult to vaccinate all the people, so which is right now is very important to vaccinate every people. So the population is increasing at an alarming rate. Production is not increasing, it is stagnant. If you look at the different productions rate, you can see that the production is not increasing. It is getting stagnant. And several problems comes out in the recent days. Increased temperature, salinity, drought, biotic stress, all these are coming and our crops are getting damaged. And our crop is unable to cope up with these new stresses because they didn't face such before. That's why traditional breeding is unsuccessful. So now, if you, being a student, you know that the central dogma of the molecular biology, that is DNA first transcribes into RNA, RNA transcribes into protein, and that protein gives you the trait. This is the central dogma of molecular biology. And you should remember this. This is very important. So if you see the plant height, because the Mendel's, according to Mendel, the plant height may be tall or dwarf, or seed shape may be round or wrinkled. So which actually derives this type of trait? This type of traits is derived by DNA through RNA through protein. So if we can change some of this DNA, we can change a trait. So this is the fundamental basis of the modern biotechnology. Now, in traditional plant breeding, what we used to do is, for example, it is a desired gene. Take any desired gene. For example, salt tolerance. It is a salt tolerance gene that is present in a traditional donor, which is not used in a commercial variety. But we have a commercial variety that doesn't have that salt tolerance. So what we did, we cross it. And when we cross it, we find the F1 progeny, which is having this desired gene in the commercial variety. But the problem is, apart from this gene, there are several other genes are also incorporated into the commercial variety. And it takes a lot of time to backcross with the commercial variety to remove the other genes associated with that. So it takes a lot of time. And also you need a very good hand of crossing. So this was the traditional plant breeding which you used to we do during our Green Revolution. Now in plant biotechnology, what we did right now is we take the desired gene from here, we isolate that gene, and then it transfers to the commercial variety. And this commercial variety only having a desired gene, only desired gene is there. So this becomes a new variety with salt tolerance. So using plant biotechnology, a single gene may be added to the single strand and it becomes a new variety. Now, what is the first product of biotechnology? Basically, in 1920s, it is already possible to isolate insulin from pancreas of pigs and cows. But some diabetic patient, they are allergic to the bovine origin of the insulin. So biochemists began to think for an alternative solution. And as you know that human genome sequencing is already known, so what they did, they identified the human insulin gene. And then this human insulin gene, they transfer to the expression vector. Expression vector is nothing but a plasmid from where the expression cassette is there. There you, using the restriction enzyme, you insert the human cDNA, which produces the insulin, and then they pass it into the bacteria, and this bacteria produce insulin. And the first insulin was product by Eli Lilly Ranbaxy, and they called it humulin N. That is the first recombinant insulin was produced, and nowadays all the insulin produced in the market are recombinant one. So this is the first success story of biotechnology, and this is the first product also. Now I come to the plant biotechnology, because we are talking about plant. I gave you example because this is the first example of the biotechnology product. But when we come to the plant, how could you make a plant with a transgenic plant? So all of you know that there is a bacteria, Agrobacterium tumefaciens. It is a bacteria which causes the crown gall disease. And why they cause crown gall? Because these bacteria have a plasmid which is called the TI plasmid. And in this TI plasmid, in the TI tDNA, there is two genes. One is auxin synthesis gene, another is cytokinin synthesis gene. So this DNA basically transfer to the plant cell. And when they transfer to the plant cell, this auxin and cytokinin gene expresses. And when they expresses, they actually induces the cell division. And when there is a uncontrolled cell division occurs, there is a formation of crown and gall. So we call it as a crown gall disease, because it's nothing but a type of cancer in the plant. So now question is, if we remove the cytokinin and auxin gene from this tDNA, then this tDNA will go to the plant cell. Now, instead of cytokinin and auxin, we introduce a new gene and then transfer to the plant cell. Now here the tissue culture appears. If you have a very good regeneration system, then from this plant cell, this transformed plant cell, you can regenerate a plant. And this plant will have a plant with a new trait. That's we call a transgenic plant. So the procedure is very simple for some plants, but for some plants it's difficult. But this is the technology. But apart from this technology, another technology is there, that is gene gun technology, that is helium gas gun technology. This is nothing but this type of instrument, and you have a gas acceleration tube, and here is a rupture disc, and you have a tungsten or gold coated DNA, and this DNA should be fired into the target cell. And this DNA, along with the gold particles, it's like a bullet. They enter into the plant cell and goes through to the nucleus, and then transfer its DNA to the chromosome and integrated into the DNA. So this technology is also there, but the problem is in this technology, maybe more than one copy number is getting attached into a single cell. So that is also another problem. And from these cells, if you can regenerate the plant in vitro, you will get the plants with the transgenic traits. Now, in the all over the world, you can find that the purple regions are the places where the transgenic plants are cultivated. In India, only cotton transgenics is allowed, not other. But in other countries, all other countries, there is they allowed the transgenic technology. Now I will tell you some story. First is the golden rice story. As you know that vitamin A deficiency is a major problem. And more than 100 million children suffer from this problem. And for many countries, the infrastructure doesn't exist to deliver the vitamin A pills. So how you can make an alternative system? So Indo Portuguese, who actually is the pioneer for developing this technology, what he did is, this is the beta carotenoid pathway in plants. You know that beta carotene is the precursor of the vitamin A. So this is the basically the pathway from which IPP from geranyl diphosphate, then phytone, then lycopene, then beta carotene was synthesized. But in rice, these enzymes are lacking. They don't have these enzymes. So beta carotene is not synthesized in rice. So what he did a daffodil gene, there is a phytoin synthase isolated from a daffodil, inserted into the rice. A single bacterial genes which perform the both enzyme and wild rice varieties have this lycopene beta cyclase gene. So he make this pathway complete in the rice. So when this pathway is complete and functional, the rice is producing beta carotene into their seeds. So the golden rice has been synthesized. And now golden rice is now cultivated in Bangladesh, Philippines, and in other countries. In India, there is a trials are running. So it is not even permitted, but trials is still running. Now, the practice of fortifying foods with vitamins such as folate, vitamin A, micronutrients like iron, zinc, iodine has dramatically reduced the malnutrition in much of the world. So this technology is available now. And with this technology, now you understand that the life expectancy is increased as compared to the 90s. You can just see how much life expectancy is there in each country, even in the developing country or underdeveloped country. It is increased because of these technologies. So as I told you that vitamin A enriched rice, that is golden rice, is there, iron enriched rice is there, and here you can find that wild type tomato, and this tomato is rich in anthocyanin, so the color changed. So highly antioxidant enriched tomatoes are there. Similarly, another technology is the antisense technology which was developed. This is, you can see, this is melon, which is actually wild type, and this is rotten after 10 days. But this is, after 10 days, it is not rotten, it is just intact. It's nothing but, you know that the ripening was controlled by ethylene, and ethylene is synthesized by ACC oxidase or ACC synthase. So if you stop producing ethylene in the fruit with the antisense technology, then you can reduce the ethylene production. Here they use the antisense ACC oxidase gene, and you can see that this melon is not rotted like this one, wild type one. So it increases its past life or shelf life, you can say. Now, another very important thing in biotechnology is that, as you know, right now the petrol is nearly 100 rupees per liter. It is increasing day by day, and it will increase. So what is the alternate source? So here comes the biofuels. Biofuels is nothing but for example, ethanol. In plants are rich source of starch, glucose, xylose, and other sugars. So if you have a technology to convert them into the ethanol, then you have a breakthrough in the research. Then you don't need petrol or diesel. Ethanol can be used as a fuel. So plant can be a good source of biofuel. And if this technology is in very preliminary stage, there are some reports that how to convert them into the ethanol is there. But in a large scale, if somebody is successful to produce the ethanol from the plant source, then it will be a breakthrough for this technology. So this is one of the areas where the research can be done in more extensive way. Now coming to another important story, that is Bacillus thuringiensis. You know this bacteria because this bacteria produces a toxin, that is a cryotoxin or BT toxin, which actually affects the insects belonging to a particular order, that is Lepidoptera or Coleoptera or Diptera. Because these larvae, they make a huge damage in crop plants. So people used to use this bacteria as a biopesticides. They just spray the Bacillus thuringiensis into their crop plants. And this bacteria protects the Lepidoptera and Coleoptera or Diptera like larvae. But later on, when the science was developed, then they identify that these bacteria basically produce a cryprotein. So they identify that cryprotein, they identify a DNA which produces this cryprotein, and then they produce, then they incorporate into the different plants, so that the plants is resistant to the insects. In India, remember this date, 26 March 2002, the first BT cotton, the transgenic cotton with Cry1 protein was approved. And this is the India's first transgenic crop that has been approved in a large scale. So nowadays, all the BT, cottons which is produced in India is almost BT cotton. They are transgenic cotton. And if you look at the graph, the production, you can see after 2002, there is huge increase of production. So that means with this BT technology, you just increase the production rate. Now another very interesting development was done by Professor Deepak Pental. As you know that brinjal is a very popular vegetables, and in India it is been used for last 4,000 years. But the problem is there's a lot of problem from insects, that is brinjal shoot and fruit borer. And this larvae or insects damage almost 60 to 70% of the crops, even after multiple insecticide spray. As you can see, this damage. This is that all damaged fruit. This damaged fruit. Here the shoot damage, and here the fruit damage. So this, they damaged extensively. So why we need BT brinjal? No natural resistance in the germplasm was there. I told you that gene pool is getting very small. So no natural resistance in the germplasm of brinjal is found. Limited success with traditional management studies and huge use of pesticides results in development of resistance in insects and also harmful effects on environment and health of the farmer and consumers. But BT brinjal has the potential to produce economic and health benefits. So BT brinjal is nothing but a genetically improved brinjal developed by inserting Cry1AC gene, that is BT toxin gene, in the brinjal genome. And this gene gives resistance against the Lepidoptera and insects like brinjal fruit and shoot borer. And when you just compare, this is non-BT and this is BT, the insect cannot infect. And if you look at the non-BT brinjal and this is BT brinjal, you can see the difference and how actually it improves the resistance. And this BT toxin is have no toxic impact in higher animals, in human particularly, because in our stomach we have acidic pH, very low pH, 1.5, and absence of required receptors. The receptors is absent for the BT toxin. So it is not at all harmful. When it goes into our stomach, it immediately degrades. So BT is very safe for non-target organism in human. And this slide I just put intentionally to show you that because there are so many questions coming out from the audience, because how you can say that transgenic technology is safe? Because when you wanted to make a transgenic technology, you have to go through several procedures. Once you develop a transgenic plants, it has to go through the IBSE, then RCGM, then review committee meeting, and it takes around, it is in the brinjal case, they developed it in 2000 to 2002, and it approves 2008 and 2009. In India, it is still under consideration because Supreme Court is still having some reservation, but in Bangladesh, it is cultivating right now. So it takes around 6 or 8 years to analyze whether this technology is safe or not. So there is the procedure which makes you feel that yes, this technology is safe. It is not like that. You develop a transgenic plant and directly put it into the field and then you start eating. No, it is not like that. It takes time. It takes several organizations, several steps to confirm that your technology is safe. So this you need to understand first. Now the second part of my talk, that what we are doing at CSI and NBRI. Now this is our website. You can go through the website and you can find that we are working in different aspects: plant diversity, systematics, Iberium, pharmacognosy, phytochemistry and product development, plant ecology, environmental technology, molecular biology and biotechnology, where actually I am working, plant genetic resource and improvement. And we have a very beautiful botanic garden. As you can see here, this is Wellestia. This is the only plant that is present in our garden in India. There is no other plants in India. So this is Wellestia, and this is cactus, so big cactus, you can very beautifully preserved here. So I invite all of you, once you come to visit our garden, you can see plenty of very important plants and endangered plants and important economical plants also. So now in NBRI, I just briefly outline what the major work we are working in biotechnology and molecular biology division. That white fly menace in India, North India, 2015, you remember in the newspaper, white fly actually destroyed Punjab cotton. And so many farmers commit suicide. So basically this white fly is a kind of fly. They lay eggs on the bottom surface, lower surface of the leaf, and pesticides never reached on the lower surface of the leaf. And the eggs are also very resistant, and soon the eggs hatched and the population comes out and they destroy the whole plants. So what they did here in NBRI, they discovered ferns, because the ferns, they are very resistant to this white fly. So from the ferns, they identify one protein, actually not one protein, 5 protein, they are resistant against this white fly. And when they identify these genes, they put it into the cotton. And you can see this is the wild type variety. And here you can see the eggs of the white fly. And this is the transgenic one. There is no white fly egg is there. So with this technology, they develop white fly resistant cotton. So Dr. P.K. Singh from our department, he actually identified that from a Tectaria microdonta fern, they identified one protein which is called TMA12, and then they isolate the gene and then make the transgenic cotton. And this transgenic cotton is resistant to the white fly. Another important thing was also discovered in NBRI. They use the same technology, antisense technology, to increase the vast life of banana. As you can see here, that after certain times at your home, you can notice that the banana becomes this type of color. And we cannot eat these bananas, we just throw it away. But with this antisense technology to block the ethylene synthesis, we can, after 9 days, when the control banana is like this, the transgenic banana is fantastic. looking fantastic. So this technology is with us. We are looking for this technology to be in the market. But in India, the transgenic technology is having a little problem. But I hope soon people will adopt this technology because the public perceptions is getting changed right now. So I hope in near future that perception will change. Another important is the increase the vast life of tomato. Using the same antisense technology to reduce the ethylene production, we can increase. This is 19 days tomato, and this is 31 days tomato. You can see the difference. So you can increase the shelf life. So that is also another important aspects. Another important aspects we are working is that in drought. As you know, that drought is a very common problem in northern India, or not northern India, even in the southern India also. Because of the climate change, because of the destroying the forests, we are just making things very difficult for us. Water level is decreasing, so it's very difficult for cope up with the drought stress. As you can see that the red zones are severely water scarcity is there around the world. So in our group, in this group of our division, they identified one gene, that is a CAMTA gene. When they overexpress in the cotton, this is the control plant and this is the transgenic plant, and you can see the difference that the root architecture is changed, because if the root is robust, the plant will be resistant to the drought stress. So here they increase the root system architecture. So changing the root system architecture, they make the plant resistant to, in case they tested it in tobacco, they tested in the cotton also. So cotton becomes a very resistant to drought stress. My lab is also working in the drought stress, but in rice. And we also identified one gene, that is a rice dehydrin gene. And when we just overexpress it, and you can see this overexpressed line is growing very well as compared to the wild type. And when we see the root system architecture, the root is getting very long. And also the architecture is also changed. So in this way, we just developed plants with drought tolerant rice. And apart from that, my own area of work is that arsenic. Because arsenic is a huge problem in India, particularly in northeast region and also in the UP region also, because Balia, Lakhimpur, Kheri, these areas are totally contaminated with arsenic. So we try to develop the low grain arsenic in our lab. But to give you a very brief introduction, that arsenic is not a new world problem. As you can see that in Chile, the pre-Columbian Chilean mummies, they have huge amount of, this is the mummy, and they have huge amount of arsenic in their bodies. This indicate that this population is actually dies because of the arsenic poisoning. So it is not a new world problem, it is an old world problem. And this pre-Columbian Chile mummies is very old, 5,000 years BC. As you can see, the arsenic found in Chilean mummy is 5,000 years BC. And after that, there are several things, and we call the arsenic is the king of poison. Why? Because maybe Napoleon Bonaparte was slowly poisoned with arsenic, and his death is because of the arsenic poisoning. So that's why the name came. It is a king of poison. Then in 1937, arsenic in groundwater was reported. Then in 1983, WHO have set a provisional tolerable weekly intake. In 1980s to 1990s, arsenic detection is rice is known, and then there is a first report of the development of arsenic resistant plant was noted in Terris Vitata, and in 2011 the first transgenic plant we developed. So as you can see, this is the worldwide the arsenic contamination, and here it is the in India, in Bangladesh, as well as West Bengal, as well as Bihar and Uttar Pradesh is totally contaminated with arsenic. And when this groundwater contaminated with arsenic is used for the irrigation of rice, they accumulated large amount of arsenic in the rice grain. And because of the rice grain, there are several chronic arsenic poisoning, as well as melanosis, hyperkeratosis, lung disease, black foot disease, gangrene, diabetes, cancer, everything is associated with this arsenic problem. And WHO set the provisional maximum tolerable daily intake is 2 microgram per kilogram of body weight. So we started working, and we started finding out that some of the varieties they we identified some of the rice breeding lines, and they accumulated very less amount of arsenic. And this particular breeding lines we called Muktasri. It has been released by the state as well as in the national level also it is released. This Muktasri, this is free from arsenic. But the problem is this is an aromatic rice. Aromatic rice, not so many people eat aromatic rice, and also farmers are also have reservations of taking aromatic rice. So we try to develop using the molecular breeding program. But the molecular breeding is very difficult because arsenic accumulation in rice is a very complex quantitatively inherited traits. So identification of a QTL is very difficult. It is already reported. So the next option is transgenic technology. So if you just look at this diagram, this is the arsenic metabolism in rice that is recently updated. Arsenic is present in two forms. One is inorganic arsenic, another is organic arsenic. Organic arsenic is not toxic. Inorganic arsenic is toxic. And in inorganic arsenic, there is two forms of arsenic. One is arsenate, another is arsenite. Arsenate enters through the phosphate channel, and arsenite enters through the aquaporins. Once arsenate enters into the plant cell, this arsenate converted to arsenite by using the glutathione or the arsenic reductase. And this arsenite then combines with the phytogelatin or glutathione and then sequester into the vacuole. So this is the mechanism of detoxification. But in some cases, they using the other transporters, for example, NRMPs or NIPs, they enters into the xylem and slowly they enters into the grain and deposited into the endosperm. So this is by some unknown mechanism. So there is a problem. So if you wanted to identify QTL, you must end up with the phosphate transporter or aquaglyceroproteins. So that is a bigger problem. That it is not related to the, because phosphate transporter is also transporting phosphate. So QTLs with the phosphate transporter will not be in use in the molecular breeding program. So if we use the transgenic approach, what can be done? So we can do one thing, we can sequester the arsenic into the vacuole so that they cannot go into the xylem or other places. We can efflux out the arsenic outside the cell. This is one technology we can do. Another one is that we can give a protective layer so that arsenic cannot enter into through this protective layer. Or the third one, we can just convert the inorganic arsenic into the organic one. I told you that organic arsenic is not toxic. So if you just convert it from inorganic to the organic one, you can just have safe rice. So this is the technology we first use for the sequestration. Sorry, I just take almost 1 hour. So can I go for another 15 minutes?
Yes, sir. Yes, sir.
Okay. So this is the first sequestration process. This is a plant called Ceratophyllum dimursum. This Ceratophyllum dimursum is basically a hyperaccumulator of arsenic. So what we did is we just take we just take the identify the phytogelatin synthase from this plant, and then we make the transgenic rice plant expressing this phytogelatin synthase, and you can find that phytogelatin is increased in the transgenic rice. And this is the micro XRF map. If you cut the sections of the rice seed and using that some technology, you can identify the arsenic content in their endosperm. And here, this is the non-transgenic and this is the transgenic. And you can find this is the arsenic is very high and arsenic is very low. And also you can find that in grain, the arsenic is reduced almost 50%. But the problem is arsenic is reduced in grain but increased in the shoot, because in the shoot they are sequestered into the vacuole. And here another problem is the rice shoot is used as a fodder. So again it's a problem because you are giving the fodder of arsenic rich food. So this technology is not very useful until unless you can restrict the arsenic into the root. So using the root specific promoter, we are doing this experiment again. Now another important gene we discovered, that is glutaredoxin. And when we knock down this glutaredoxin in rice and overexpress the glutaredoxin, then we find that knockdown plants under arsenic stress is sensitive, but the overexpression line is very tolerant. And when we just see the arsenic accumulation, we find that knockdown plants accumulated higher amount of arsenic, but the overexpression line accumulate lower amount of arsenic. That means they somehow efflux out the arsenic from their cells. So here you can see that this plant overexpression lines, the panicle is very good in condition, but when we give the arsenic treatment, the panicles is very small and low production. And also the rice, this content huge amount of arsenic in their grains as compared to the transgenic one. Now, what happened here? In these transgenic lines, basically what happens when we overexpress this gene, this gene actually increase the production of LSI1, that is an aquaporin gene, and decrease the NIP 3.1 transporter. So what is the role of LSI1? LSI1 takes the arsenic into the root cell, and NIP 3.1 is taking the arsenic to the xylem. So when we overexpress the gene, we just stop the plant to go arsenic into the xylem. So it restricted into the root cell. So if you look at this picture, you can see that in the root, the level of arsenic is quite higher, but in shoot or seed, the level of arsenic is very reduced. So in this way, we just reduce the arsenic in the seed. Another important approach, that if we can just make a coat of protective layer so that they cannot go, arsenic cannot enter into the plant cell. So we identified one class 3 peroxidase gene, and this class 3 peroxidase gene, when we fused with the yellow fluorescent protein, we find that this gene present in the cell membrane or the apoplast region. That means this gene is actually present in the apoplast region. When we overexpress this gene in the Arabidopsis, what we find that upon arsenic treatment, this plant is getting resistant. And this is because this peroxidase gene produce a lot of lignin. So the roots are having a lot of lignins as a protective layer. So the arsenic cannot enter through this lignin layer into the cells. So the arsenic level is reduced in the model plant Arabidopsis. So this is a good technology. Now this technology we wanted to transfer it in rice. And this is the another technology which I said that is very important, because if we convert the inorganic arsenic into the organic arsenic, then you can reduce the toxic effect. And here in this case, we identify one arsenic methyl transferase from a Ascomycetes fungi. And this gene, when we overexpress in Arabidopsis, we find that yes, there is a reduction of arsenic in their plant. We transfer it into rice. And what we find, we do the pot experiment, and we find that non-transgenic plants, when exposed with arsenic, they grow like this. And these transgenic plants grow like this. So you can find the difference between the two plants. And if you find the difference between the panicle production, you also find the difference. And here you can find that the seeds contains very less amount of arsenic. Why? Because most of the arsenic converted into the organic one. And this organic one is volatile in nature. They are actually trimethyl arsine, which is volatile in nature. So this gene basically converts the arsenic into a volatile compound. So it just comes out from the plant. So this technology is one of the best technology for the productions of less arsenic in the grain. So this is the future plan. that to how to use this volatilization, because we need to test into the field first, and also the toxicity and all these things. We are also working with the gene editing technology. We are also working with the microbes, because some of the microbes, if we grow along with the plants, they reduce the arsenic uptake via root system. We wanted to block the arsenic mobilization from root to shoot by knocking out some specific gene. We wanted to identify some other minor QTLs which regulates the arsenic accumulation. Here we started working on the gene editing technology. As you can see that this is the gas plant. This gas plant is when we just knocked this gas line, there is no gas or little gas is there. So we knocked the gas reporter gene using the genome editing technology. Now we targeting LSI1, LSI2 or different transporters to knock it out, so that the arsenic cannot go from the root to shoot or to the grain, and so that we can get a plant with the desired phenotypes, because genome editing plants doesn't require the transgenic technology approach, so we can directly market it to the field or to the farmers. So you wanted to use this gene editing technology in our research. This is some of the photographs that our transgenic approach with methyl transferase by biovolatilization has a great work, and for this work we have been awarded by Gandhian Young Technological Innovation Award in 2019, both as a guide and as a student also. And this is my whole team. Some of them are alumni. They are in a different country right now, and here is the current batch of students who are working tremendously in gene editing or in other characterization of other genes. And I really thank all of my students for their wonderful work, and I also thank you for your kind attention. Thank you.
Thank you, sir. It was definitely a wonderful lecture, I will say, and it was very much helpful for our PhD scholars who are here, and also the MSc third semester and fourth semester students who are listening to you. They must have been greatly benefited by it. And I will take just 5 more minutes will be given. And if there is any question, please unmute yourself. A relevant question to be asked to the guest, please, you can ask the question. So, I think they have all understood you well, and yes. So it was as you said. I just came to my mind that as you said, there is the various plants have been approved, like the BT cotton and other plants, the variety of rice, golden rice. What is the major problems are coming across by in not accepting it and not allowing it to grow?
This is actually a policy taken by the government.
Huh.
Now there are several problems are there. First problem I find it's because of the transgenic plants, the lot of pesticide industry will be affected. So pesticide industry will not want that the transgenic plants will prevail, because they will be in a huge loss. So you understand that is a political game is playing there. So that is there, but people should understand that pesticides is not good for your health, because if you go to the market and have a very good brinjal, bring it to your home, I'm sure that is having a pesticides inside. It is better to have the brinjal which is having a board, which is affected basically, because at least you want that brinjal is not given the pesticide treatment.
Yes, sir.
So I think that government should take, and also there is a need to educate people that transgenic is not harmful, because people have a strong mind that maybe the transgenic plant will actually is a very harmful for the health or for the environment or something like that. But it
Yes,
is
sir,
not like that. So
you are,
one should understand that. So educate people that no, it is not harmful. It is useful technology. So adopt it. So
so
that's
yeah,
that
I
is
think
my
you have answered the question.
yeah.
And it is a big industry of pesticide, we all know, and they increase the shelf life so later on, and initially also when I have gone to Punjab and different farm area, and I've seen how much pesticide is there, even taking the topsoil and even the A horizon soil, the pesticide is percolating there. So it must be a big industry, and they must have some hindrances. And but I want to thank on my behalf, and later on I'll give it to
I have
Deepa
also
ma'am.
one question, Tulika. Please let me ask you.
Yes, ma'am. Please ask questions.
Sir, you have taken CRISPR technology, this CRISPR-Cas. Are you
Yeah.
utilizing this in your lab because it is the latest technology? Are you utilizing it in plant breeding or tissue culture approach?
Yeah, I mean, tissue culture is must. You know, tissue culture is a must for any technology to incorporate. Yes, we are developing this technology in our lab, and we developed already. We now have our CRISPR lines, I mean, gene edited lines already that has been developed, and we have lines. I could not show you the results because this is very, I think it is a little bit early to show. So that's why I just give you an idea that yes, we started, but already we have the CRISPR lines.
Sir, CRISPR line is utilized for virus-free plant, and you are utilizing it for molecular breeding.
Yes. We are just no. We are just targeting some transporters. We are just knock it down.
Okay, sir. Thank you, sir.
Okay. So I mean, students, if you have questions, then must be you can ask me through my email or something like that. So no problem. I am not a very that kind of guy who cannot who will eat you up. Don't think like that. You can ask any questions, no problem, because asking questions will actually increase your knowledge. I mean, maybe no, maybe I cannot answer all the questions, but I can try.
So thank you, sir. And I am definitely know that students want to ask, but they will ask. You have said they can ask through the email. And for my behalf, and you're also co-guide with me with my two students, Archana and Pallavi. And thank you for accepting that.
They are working also.
Yes, sir. So now my colleague also, Deepa ma'am is here to express the vote of thanks. Please, ma'am.
Thank you, Tulika. Good afternoon to everyone. I am Dr. Deepa Srivastava, and I am feeling honored to get this opportunity to give vote of thanks to today's event of 7-day workshop. Certainly today's lecture was very meaningful and fruitful, and we have got some recent knowledge about plant biotechnology and its application. Now it is the time to convey our gratitude to all who have extended their support to make this event successful. We would like to express our deep sense of gratitude to our Vice Chancellor, Professor Rajesh Singh, for his blessings. We most humbly thank our Dean, Science, Professor Shantanu Rastogi, and Senior Most Professor, Professor V.N. Pandey, for his presence itself is a motivation for us. We are thankful to Professor Pooja Singh, HOD, Department of Botany, for providing his support and making this event possible. We extend our thanks to Professor A.K. Dwivedi for providing his wholehearted support in this workshop. Our words are not capable to express our sense of gratitude to our speaker, Dr. Devashish Chakrabarti, Senior Principal Scientist and Tissue Culture Head, NBRI, Lucknow, who has given us insight about BT cotton, arsenic metabolism, transgenic approach, molecular breeding, CRISPR technology, and many other things, cryogene, many other new things. I am sure students have learned a lot and they can ask many questions. Even he is very helpful person and he support us. Thank you, sir, for providing your valuable time. Your each word are important and meaningful for us and our students. We are very grateful to our faculty members and our colleagues, Dr. Tulika, Dr. Shweta, Dr. Rajvi Singh Chauhan, Dr. Virendra Kumar Madhukar, Dr. Rakesh Pandey, Dr. Spriti Mal, Dr. K. Sunita for providing their time for shaping this program so nicely. We have been fortunate enough to be backed up by a team of very motivated and dedicated students, research scholars, and a staff member. We are thankful to all of them for their contribution. My deep sense of gratitude to organizing members for their effort and for shaping this event. Last but most important, I am thankful to our students who were patiently present here. Thank you all for providing your time this afternoon. Thank you so much. Over to Dr. Tulika.
Thank you.
Thank you, ma'am. And with this, I will again thanks the students and end this lecture. Thank you, sir, for coming here.
Thank you.
Thank you. It's my honor. Thank you very much.
Thank you, sir. Thank you,
Have a nice day.
Thank you all. Now we end this meeting. Okay. Thank you so much.